There's so many positive alarms. And then there is electrons. These electrons, because the electrons are so many electrons. So this positive ions, there with these electrons can delocalize themselves around. This metal is held by a strong forces of attraction. This metal
is wrong.
ions.
Good morning, everyone.
So this is a metal that has so
Hello.
many positive ions. So when
Good morning, everyone.
around,
Please, is there a course rep for this course?
this kind of bonding, this kind of
Hello.
Hello, good morning,
Hello.
ma'am.
Is there a course
morning,
rep
ma'am.
for Zoo 101? Good morning.
Ma'am, the project for medicine and surgery, but Professor Fisher has said that it's the hosting now, ma'am.
positive ions, there with these electrons can
Okay, I just need somebody to help me administer the class. So many background noise.
Oh, all right.
Yes, so who is speaking now? Who is speaking?
I'm Temidayo, ma'am.
Okay, so Temidayo,
Yes,
well,
ma'am.
you are the host. Can you make me a co-host? And then can you help in muting people? Can you mute everybody and only unmute me?
When these electrons are moving
Will you get me? All right, so everybody should remain muted. Thank you very much, Temidayo. So everybody should remain muted while I only should be unmuted. If anybody has any question or comments, they should raise their virtual hand. If I acknowledge you, then Temidayo will mute you, or you post your question or comment in the chat box. Now, please ensure that you rename your device with your name and your surname. And Temidayo, please make me a co-host because I'm going to remove anyone whose name is not written here. I can see somebody with ... I don't know who that is. So please, no videos on, no audio on. Fine. Secondly, please put your name, your first name, your surname, your matric number on the chat box. Put it there and your department, very important. That's going to serve as the attendance for the lecture today. Temida, do you have an idea of the number of students that are taking this course?
I'm not sure of that, ma'am.
So in your normal classes, how many people have been attending the courses?
So like it's currently linked to YouTube now. So like the total subscribers are usually like 400 plus.
YouTube, which YouTube? Who is the owner of the YouTube?
It's actually mine, ma. Because the Zoom is actually limited, ma. So it's limited to 100. So to allow a lot of people to be able to connect. So that's why we linked it to YouTube, ma.
Yeah, so and is that open access? Or how do the others get that?
Yeah, I've also sent the link. As I sent the link for the Zoom, I also sent the link
Is the
for
YouTube
to them.
open access?
So it's currently like,
excuse me. Is the YouTube open to
it's open access.
everyone apart from the students?
Yes, ma'am.
No, that's not supposed to be.
Yeah.
That's not supposed to
No,
be.
it's only the student currently. It's only like the student that I sent the link to. It's not like just open.
Yeah, but if I go on YouTube now and I type your name, I will have access to this course.
So until I send you the link, ma.
Okay, anyway, I will just take it as it is. This is the only class I have with you. You should not be so. Students should even not be the one administering this. It's very, very wrong. Okay. So I just wanted to have an idea of how many people. So you said 400. So if 400
Yes.
people are taking this course and we are about how many now? We're about 100. That's a quarter of the class. That's not good enough. So what's the average number of students who attend the class when you take it? When you take the
No, like
101?
I'm saying that most of them would have joined the YouTube now, ma. Like they're on YouTube now.
Okay. They didn't log in.
Yes, ma'am.
Okay. All right.
Yes, ma'am.
All right. So let me I'll put on my video shortly, just so that you can view me. For those of you who don't know me, excuse me. I'm sure the zoology students already know me as I'm the zoology course advisor. My name is Temitope Shogbamu. I'm a lecturer in the Department of Zoology, and I'm teaching this topic on muscular tissues here in year one for you all. You probably still meet me in second semester for some of you who will be part of the course. We'll be taking the course. I guess the whole of biological sciences will still take our courses in second semester. And then for those of you who are not going to College of Medicine, you will still see me or get to know more about me when you meet me in the courses from year two going forward. So basically what I'm going to teach today within the next, less than an hour, what we have is not so much to talk about. Is we're going to talk about muscular tissues. And I like my class to be interactive, but because of the nature of this class, I think I will keep much of the questions to maybe at the end of the topic or in the course of the topic as I'm teaching. If you have questions, you have comments or clarification, you can put in the chat box. Hopefully I will see it, or you can raise your virtual hands when the time comes. Okay, so I will start very quickly with the slides and we go ahead. Okay, so if this was a physical class, usually I would start with asking the students what you have learned so far in this course, just to be sure that we're all on the same page, and to be sure that we have all been learning and I'm building on what you have been learning so far from the other lecturers in the course. Very, very important. So so far, I will just do a brief that we have been learning about the mammalian body, right? So first of all, what's a mammal? So you know that first mammals are vertebrates. They are animals that, of course, the Homo sapiens, as the human beings, belong to. And they have those characteristics of living organisms that both plants, animals have, but there's some peculiarities that mammals have, both as an animal, but also both as as animals belonging to the chordata, and then animals that are within the vertebrate family. Now, in the mammalian body, there are different organs, as I'm sure you have learned, or I think by year second semester, you'll be introduced to the progression and levels of organization from the unicellular protozoan animals to the multicellular animals, and then the organization of animals there. So we are coelomates, so you progress from those that have coelom, those without coelom is coelomates to pseudocoelomates, and then when you get to the vertebrates, we're talking about animals with coelom. So very quickly, part of the things that we have within the mammalian body is that, as we progress from cells, when we have two or more cells coming together that perform a particular function, we call them tissues. Similarly, when we have two or more tissues coming together to perform a particular function, we call them organ. And ditto, the organ, coalition of different organs, makes up the individual or system. Now, here we're talking about muscular tissues. So it therefore means that we're basically talking about different types of cells that all work together to perform the function of providing the musculature, making of the musculature, but also providing muscular functions in the body of a typical mammal. And so I'm sure that we're building up on what a lot of you have learned in secondary school. I'm almost sure that this is not the first time you're hearing about muscular tissues. So by way of objectives at the end of this topic, and if you like, you can convert them to ways that typical questions could be asked of you in the exam, which is going to be computer-based test, so it's going to be objective. So I want you to, Munira, or somebody is writing on my slides, please don't do that. Otherwise, people will not see. So please pay attention to some points as I go through the slides that I will highlight, which are things that, for example, some certain names, some certain definitions, some certain structures, some similarities and differences between the different types of muscle tissues in mammals. These are things that you need to pay attention to, because those are the ways that we could actually ask you the questions in exam. So what are the learning objectives for this topic? First, I expect that all of us should be able to distinguish between the three types of muscular tissue that we have in typical mammals. So, of course, I will introduce you to the types of muscles and muscle tissues. I will introduce you to the peculiarities or characteristics or features of each type of the muscles. And then we will see how similar they are across the three and what are the contrasts or differences across the three types of tissues, muscular tissues in the mammals. Secondly, I expect that we will be able to describe how these three types of muscular tissues differ in both structure and function. So when we're talking about structure, we're talking about the anatomy, right? We're talking about the morphology. How do they look like? So when you are given a typical muscle tissue in the lab, how do you differentiate physically by looking at it physically, either physically with your eyes or with a microscope? In terms of the structure, anatomy, or morphology, how do you differentiate between the three types of muscular tissue? But also in the function. So the three types of muscular tissues all work to perform different functions, and that also aligns with where you also find them in the mammalian body. So you should be able to describe those differences in structure and function of the three types of muscle tissues. And then finally, I expect that we should be able to learn how these muscular tissues are organized. And if you were to draw this, for example, or you were given a structure of the different muscular tissues, you should be able to identify them based on the way that they are organized. So the first thing that we want to start with, and so I'm going straight into the introduction, is to tell us how muscle tissues are derived. How do they come about, right? Where do they derive from? So you have in the human body, for example, a typical mammal. So you have the embryo, you have male gamete, female gamete, they come together, they form a zygote by fertilization, and then after fertilization, they go through different stages of cell divisions. We call them cleavage. And after cleavage, they go through all stages of division that the embryo grows. And then when it gets to a certain stage, which you will learn about when we talk about histology, that's not the focus of this, and embryology, the animals begin to differentiate. Like I talked about, we start off with simple cells, and then they begin to differentiate to perform specific functions. So they differentiate to form tissues of the eye, you have tissues of the brain, you have tissues of the kidney and all of that. So here we're talking about muscular tissues or muscle tissues. So they are of mesodermal origin. As you know, we have the three body layers, main three body layers. In a typical mammal, we have the ectoderm, which is the external body layer. We have the mesoderm, which is in between, and then we have the endoderm. So our muscular tissues generally, they originate or they derive from what we call the mesodermal layer, from the mesoderm. Now, it's very important that please you take note of this, because the question could in test or exam to identify or to select from the options provided for you from which body layer does your muscular tissues derive from in mammals. So learn and know that they're from mesodermal origin, or they are of mesodermal origin, and they differentiate by process of lengthening and simultaneous synthesis of myofibrillar proteins. So myofibrillar proteins are major components of our muscular tissues. Synthesis means formation. So as the myofibrillar proteins are being formed, then as simultaneously or in parallel, these are being lengthened and they differentiate into the different types of muscular tissues that we have. And so the simplest unit of your muscle tissue, which is your cell, is known as the muscle fiber. Muscle fiber. That's the simplest unit of a typical muscle tissue, muscular tissue. And this muscle fiber is surrounded by basal lamina. That basal lamina is called an endomysium. Please, I want you to take note of that. You will see that I put muscle fiber in capital, I put endomysium in capital. The way I could ask you in the exam is to put muscle tissue and tell you to identify what component of it of muscle fiber, the muscle fiber is, what component of a typical muscle tissue. Or I could ask you that your muscle fiber, is surrounded by a basal lamina that is called what, or known as what? Is it ectomysium? Is it mesomysium? Is it endomysium? Is it musclemysium? So there are different ways that these can be asked. So except you pay specific attention, and not only to the words or to the notes, but also to the spellings. Very, very important, because it can also be endo, and then we put endomycelia or something. So as you're learning the text, please learn the correct spellings as well. They're very important. So summary of this slide: the basic unit of your muscle tissue is your muscle fiber, and this muscle fiber is surrounded by basal lamina that is called, or also known as, the endomysium. And then the plasma membrane of your muscle tissue is known as your sarcolemma. Plasma membrane of a muscle tissue is known as sarcolemma, while your cytoplasm is called sarcoplasm. Please know the difference between both of them. The cytoplasm in your basic muscle fiber is called sarcoplasm, and then the plasma membrane is called sarcolemma. And then we have something that are called myofilaments. These myofilaments are cytoplasmic contrastile elements. So they are elements that are within the cytoplasm or the sarcoplasm. They are called myofilaments. Myofilaments. So there are many cells, of course. By definition of a tissue is a composition of two or more cells, is it not so? From our simple definition that I gave us earlier on, that your basic tissue is a composition of two or more cells, but they come together to perform specific function. So there are many cells that are made up of your typical muscle tissue, and there is just little extracellular materials between these cells. And this muscular tissue is well vascularized. What does that mean? It means that it has a lot of blood vessels that are supplied within that muscular tissues, and the cells of your muscular tissues are elongated. You will notice by the time we begin to see the structure of the three types of muscular tissues, how the composition of those tissues and how the cells are arranged. And so these cells, like I mentioned earlier, contain that myofilaments. And these myofilaments are contractile proteins. That's a component of the myofilaments that you find in the cells, which are in the sarcoplasm. So what are the three main types of muscular tissues that we have? The first one, we have skeletal muscles, meaning that, of course, when we talk about skeletal muscles, we associate them with the skeleton. We have the cardiac muscles, and from the name cardiac, it means that it has something to do with the heart. And then we have the smooth muscles. So these are the three main types of muscular tissues that we have. We have the skeletal muscles, the cardiac muscles, and the smooth muscles. Now, Very quickly from here, we can already differentiate or see similarities across the three muscles. The first similarity is that one similarity between your skeletal and cardiac muscles, as we'll see in the diagram that I will show in the next slides, is that the fibers that are within these muscles are striated. They are striated, right? While in your smooth muscles, they are non-striated. So striated means that they're sort of lined. That's what it means. So the muscle fibers that are there, the cells that are there, they are striated, while in your smooth muscle, they're not striated. So if I were to ask you that in the exam or in the test, in the OBJ, to identify or to pick which of these three types of muscles is non-striated or has fibers that are non-striated, you should be able to identify them. So that's one similarity between skeletal and cardiac, and then one dissimilarity or contrast between the three types. So only smooth is non-striated, while the other two have striated fibers. So we start off with our skeletal muscles. And here on this slide, I have here a typical structure or diagram of the skeletal muscle. I want you to take note of the structure. Remember, we'll talk about structures earlier on. Take note of the labels in the diagram. Take note of their locations and take note of their function. Because one of the things that I said in our learning objective is that you should be able to differentiate between the structure and the function of the three types of muscles. So starting with the smooth muscle, we can see that we have elongated cells. So we have here the nucleus, right? We have the striations. We can see that in this case, we have transverse striations or like horizontal line. And then we have the nucleus, on the cells, which are actually arranged perpendicularly. This is not, the way this diagram is not as if the nucleus are just placed anywhere. This is the structure and arrangement of your typical skeletal muscle. So you have very long rod-like cells. So these cells, these white ones here, and I'm hoping you can see my cursor as it's moving, are actually the cells. So the cells are rod-like, or if you want to call it cylindrical. They are long. They have striations. So in the tissue, we have striations, which are the horizontal or transverse lines. And the nucleus in the cells are arranged perpendicularly. I'm sure you understand what perpendicular means. So you have this nucleus here, and this other one is facing the other side. It is not on the same side. And you have that all across and the way the nucleus are, staged, or arranged in the cell. So striations are perpendicular to the axis of the cell, and there are several nuclei that are present on the periphery of the cells. So typical thing here, you have your rod-like cell, you have the nucleus, you have the striations in your typical muscle, skeletal muscle. So where do we find the skeletal muscle? Like I said earlier, and even from the name, your skeletal muscles attach to and they cover the bony skeleton in your typical mammals. So if you've ever, maybe gotten the bone of a cow, or you've even eaten it, you're the meat that is attached to the bones. So those tissues there, that meat there actually are skeletal muscles. They attach to it and they cover the bone, And these skeletal muscles, we also find them on the face, right? I don't know if you have ever seen pictures of maybe somebody who is sick and, there's hardly any muscle left on the face. It just looks as if it's all bone and then the bone is covered with flesh. You can literally see the structure of how the skeleton of the person is because there's hardly any muscle left. When you just hold the flesh of the person, it's as if it's lifted up. Right. So it's the skeletal muscle that provides that, firmness. Some of us are quite endowed, especially facially, we're fat and all that. So the skeleton is there, but the skeletal muscles, sort of cover and attach to that skeleton and give us that fullness or that structure, which our flesh now covers. So it's at the skeletal muscles attach to the skin. And what do they function in? The first thing is that they help to generate heat and to maintain body temperature. Remember that mammals are homoitemps. That means that we have the ability to be able to maintain a constant, to regulate our own body temperature. So your muscles help to generate heat. And that's why when I'm teaching this in histology, I usually give these examples, which rings home. And thank God quite a number of you are going to College of Medicine. Now, if you are in a cold place, right? Or maybe you go to, during winter, you go abroad to one of the temperate countries. The truth is, the fatter one is, the lesser it will be more difficult for that person to quickly get cold than someone who is quite slim, right? So someone who has a lot of muscle, people will call it flesh, but a lot of muscle, that person, is able to generate it and keep him or herself warm faster than someone who is slim. And so most of the time, somebody who is quite slim, doesn't have a lot of flesh, needs to wear a lot of, winter clothes and a lot of covers to ensure that the cold doesn't get into the person's bone or skin, because the cold can really be so bad, it can get to your skin. But someone that has a lot of muscle, it will take a longer time for the person to feel the cold. And it's the same thing. Conversely, when you are in a tropical environment like we are in and it's dry season, someone who is fatter would sweat much more than someone who is quite slim, because of the very same reason. The muscle helps to generate heat and to maintain body temperature. Then another thing is that even from the location that I gave you, you can already decipher that it also maintains body posture. Right. It helps to maintain body posture. Can you imagine a skeleton walking along on the road? It's like having sticks put together, maybe just put together by inches, which is what your joints do, to attach bones to the other. And that skeleton moving on the road. What it means is that once something touched that person, that skeleton, and the thing falls, it can literally break. But your muscle provides structure and posture and helps the person to be able to stand and all that. Who are these writing on the screen? Can you please stop doing this? Right. So two basic functions help to generate heat, help us to maintain body posture, and we find them attached to and covering bony skeleton, and also covering, they form the facial muscle. That's skeletal muscle. And in the slides, when you see this in the lab, this is what they would typically look like. So you have your cells, you have, the rod-like structures, and then you have the striations, that are here in your typical skeletal muscle. Now, the next one is a cardiac muscle. Remember, I said that the fibers or the muscle fibers of our cardiac muscles are striated. So your cardiac muscles, from the name, are muscle cells that are found only in the heart. And if you notice the difference even in structure, you will see that your skeletal muscles is elongated, is square-like and all of that, while your cardiac muscle is sort of irregularly shaped. Right. If you also look at the location of the nucleus, you will see that the nucleus are, distributed sort of like unevenly. And then we have something in the cardiac muscle, which we do not see in the skeletal muscles. And you would also see that we don't see in the smooth muscle. And that's what we call an intercalated disc. So it's also cylindrical. So you have long cylindrical cells in your cardiac muscles, and then they are splitted into small branches like trees, like branches of a tree. They have striations as well, like your typical skeletal muscles. They have the nucleus, they have the cytoplasm, which we call the sarcoplasm in our muscle tissue. But the difference between the cardiac and the skeletal, as well as the smooth, is that there is a presence of intercalated disc, which are these white things that are here in between. And there are also one or two nuclei that are present in the center of the cell. While we said in the skeletal muscles, the cells, the nucleus are arranged perpendicularly in the cell. So we find them in the heart only, and their function is that they perform the function of contracting. So the muscles help to squeeze blood out of the heart and propel them into the blood vessels. So if you touch your heart, you will see that it's pumping. What is helping it to pump are those muscles that are present in the heart. If you have ever watched movies, especially medical type movies, and you see maybe they're doing a heart transplant or something, and even when they remove the heart out of the person, you see that for a while, the heart can still be beating. Right. So all those things that, that's helping that beating that is happening actually is a contraction. It's actually pumping blood, right? Pumping blood, getting oxygenated and all that. So it's the muscles that enable that to happen, that enable that contraction. So that's the main function of your cardiac muscle, which is present in the heart, which is the work of, pumping or contractions. And this is what they typically look like when you see it in the slides in the lab. And the next and final one is our smooth muscles. In structure, you can see that the three of them actually are quite different structurally, even though we can see similarity. For example, one similarity that you would notice in terms of the structure is that, of course, they all have cells. They have our nucleus in the cell. But in terms of the arrangement and positioning of the nucleus, you can talk about that as a dissimilarity. The other thing is that the cells in our skeletal and their cardiac muscles, they are rod-like, right? They're cylindrical. But if you see these ones are like sickle-shaped. They are spindle-shaped cells that we find in a smooth muscle. So that is a difference that we find between the structure, please note that, of the smooth muscles compared to the structures of the skeletal and the cardiac muscle. So the cells here are spindle formed or spindle shaped. There are no striations. As we will notice in the other slide, we have striations, horizontal lines, as you may call it, but we can't see any striations here. They're smooth in your smooth muscles. And here, in each cells, we only have one nucleus for your smooth muscle. Meanwhile, when we saw the cardiac cell muscle and the skeletal muscle, you will see that, each cell had quite a number of nucleus. In your cardiac muscle, the nucleus was positioned in the center. In your skeletal muscle, the nucleus were positioned perpendicularly to each other. But here in your smooth muscle, there's just one nucleus, which is positioned in the center of the cell. There is no striation here in your muscular tissue. And we find them lining the walls of ducts, the walls of blood vessels, the walls of lymphatic vessels. We also find them lining the walls of our digestive tracts, the respiratory tracts, and the urinogenital tracts. We find them, of course, urinogenital in the scrotum, in the penis, in the perineum, in the eye, in the skin, and in the nipple, that is the breast area. Now, what do they function in, the smooth muscles? They help to propel contents along the tube. You can typically get this example when you look at your digestive tract. We start from your throat area and then all the way to the large intestine. So when you eat, you put your food in your mouth, your tongues help you to transfer this into your throat, and then it begins to go. In fact, you can literally feel it sometimes as your food goes through your digestive tract. What enables that to happen, or what helps that or facilitates that propulsion of your food, is the smooth muscles that are lining the digestive tract. And that's why you see sometimes maybe you eat a bone or something, it's quite obstructive because it's a smooth muscle, it's a muscle. So it can actually literally be punctured. So sometimes what do you do when you eat a bone? You try to get a bar or bread or something to help you to support that bone to go smoothly with obvious, or hopefully not causing any abrasion as it propels through your digestive tract into the stomach where it's digested, and then the excretory products pass through from your large intestine and comes out through your anus and all of that. So they help to propel contents through the tube, it surrounds, as you also find in the intestine. The second function that they do is that they regulate the amount of fluid flowing through it. For example, as in the blood vessels, they help to regulate the amount so that the amount is not too small, is also not too not too much. Right. Sometimes you hear that people have blood clots and all of that. And so they take drugs or medications that are sort of like thinners. They help to thin the blood so that it's easy for the blood to flow through the blood vessel. And the muscle is able to facilitate that flow of blood and it not clogging and then clotting, which can actually be a problem. So note the differences in functions across the three types of muscle tissues. Remember that our skeletal muscles, they help to maintain body posture. And then what also do? They help us to generate heat. While your cardiac muscles serve the main function of contraction, right? They help to contract and then allow blood to flow in through from the heart into the blood vessel and all of that. While for your smooth muscles, they help in propelling contents down the different tubes, your digestive tract, your urogenital tract, your respiratory tract, and they help to regulate the amount of fluid flowing through the blood vessel. So that's some difference that we find in the function. In the structures as well, we have talked about the differences across the three types. We said our skeletal and our cardiac muscles, they have what we call striations. We said the cells that are present in our cardiac muscles have multiple nuclei. Only difference is that in your skeletal muscle, the nucleus are arranged perpendicularly to each other in the cell, while in your cardiac muscle, the nucleus are at the center of the cell. We also said that the cells of the skeletal and cardiac muscles are rod-like. While they're quite straight in your skeletal, they are branching in our cardiac muscles. While for our smooth muscles, there is no striation at all. The cells are spindle-shaped and only one nucleus is located in the center of the cell. And this is what our typical smooth muscle looks like. If you look at it, you will see that even in the structure, as I showed us earlier, they're a bit different to the way we saw for our cardiac and our skeletal muscles. And that's all. Like I said, it's quite short, basic things that we want to learn for your level year one. What are the three types of muscles? What are the basic components or structures of your typical muscle? What are the differences and similarities in structure and in the function across the three types of muscles that we have? And then what are their locations? Okay, so that's the end. Is there any question or comment? Whoever is loading whiteboard, can they stop this, please? Any question? I'm ready to take up questions. We still have a lot of time. This is just 10:37. Do we have any questions? You can raise your virtual hand. Okay, thank you. Monira, Sonny, you can unmute. Let me enable you to.
Good morning, ma'am.
Yeah, morning.
Yes, ma'am. In the cardiac muscle, is it that is in each cell that we have the nucleus base at the center, or in different cells, like it's just scattered based on the center?
No. It's each cell. Let me open it up again so that we can speak to it. Okay, so I'm hoping you can see my screen. Yes. So, like for example, can you see my cursor moving?
Yes, ma'am.
Now, so this is the tip of one cell, and this is the end of the other cell. And so in this one cell, you have this one nucleus there in the center. This second one too starts here and ends here. You have your one nucleus there. So it's each cell. Because you remember we said when we were in the introduction, we said that the cells are together and there's little extracellular material between them. So that's why they look like they are quite joined together. There's hardly any space between them. So if you don't look at it well, you may not be able to see that they're individual cells. but they're all joined together. And so in each individual cell that you have the nucleus at the center. Yeah.
Thank you.
Yeah. Okay. Let me see. Do we have any other hand while I check the chat in case there are any questions in the chat? Do we have any other? If you have any other question, please feel free to unmute. Or let me check if I can recognize you. I can't see any question in the chat box. Any other question? Clarification? Because if you don't have, I have. So we ask you just to be sure that we're all on the same page here. Ma, check YouTube. Okay, I don't have access to YouTube. The person who has the account, I don't know, where it is, so I'm unable to. I suppose you mean that there are questions on YouTube. I can, if anybody can access it, they can please post it here. I can't access it from here. Ma, where did you say the nucleus and skeletal muscle is located? Yes. So in the skeletal muscle, I said the nucleus is located within the cell, but they are perpendicular to each other in the cell. So this is our skeletal muscle. This rod here, from this beginning here to this here, is the cell. And these are the nucleus. And they are arranged perpendicularly to each other in alternate rows, as we can see in each other. However, when you go to your cardiac muscle, you can see that they are rod-like. These are each cells, and the cells are located in the center, just as we had for your smooth. Only that in your cardiac, we have striations. In your smooth, we don't have striations, and we only have one nucleus in the cell in your smooth muscle. I hope I answered that question. Omorishola Ogumekon. Okay, Anastasia, please, ma, can you come over from the beginning before the types of muscle? Okay. All right, so let me quickly do that for Anastasia, I guess. Yes, so the beginning, what I was doing in the introduction is to basically give us an overview of the main components of your typical muscular tissue and where they originate from, where you find them from. And this was where I was saying that, in just these 2 slides, I can ask you almost 20 questions. So that's why I said pay very much particular to each word on this slide. I said our muscle tissues derive from the mesodermal layer. So they are mesodermal in origin. And they differentiate through process of lengthening and synthesis. And I said synthesis is the same thing as formation, right, of myofibrillar proteins, which are the components of your muscle tissue. I also said that the basic unit of your muscle tissue is a cell, and that cell is known as a muscle fiber. And that muscle fiber is surrounded by a basal lamina that is called an endomysium, right? And the plasma membrane of your cell is called a sarcolemma, while it is cytoplasm is called the sarcoplasm. And inside your muscle cells, we have contractile elements that are called myofilaments. They're called myofilaments. And I mentioned that the cells are quite close together, as I showed you in the structure of your smooth muscles. But even as you would see for your skeletal and your cardiac muscle, they're quite close together because there's little extracellular material within them. So there's little space between the muscle cells or muscle fibers, and that these muscle fibers or cells are well vascularized. So the tissue is well vascularized, it's supplied with lots of blood vessels. The cells are elongated. As we saw in our skeletal and cardiac, they are rod-like or cylindrical, while in our smooth, they are spindle-shaped, but they are elongated. And the cells contain myofilaments, which are contractile proteins. So those are the main points that I put forward there before we went into the type of tissues. I believe that answered that question, Anastasia. Ma, so sorry, I was the one who mistakenly loaded the whiteboard. Oh, that's fine. Let me see. Temi Lokwe. Okay. Okay, Ma, please, can you explain what myofilaments are in relation to histological characteristics of the muscle? Yes, so basically, actually, the slides I'm showing you are the slides that I used to teach histological characteristics or description of the muscles. So your myofilaments are actually part of the histological features. So what do we mean by histology? We are talking about when you cut through a section of the muscles, what are the components that you see there? What are the contents that you see there? How are they arranged, right? How do they look like in structure? That's what we're looking at. And that's what we talk about when we talk about histology. And then, so that sectioning, we usually view them under the microscope. So what we're showing you, all these details that we're talking about are things you would usually only be able to see under the microscope. And sometimes when you're even going to very minute details as your myofilaments or contractile proteins, you will need to go further to be able to see with an electron microscope, for example. So that's basically what I've talked about, the myofilaments. They are contractile proteins that make up the cells of your muscles or your muscle fiber. I hope I answered that question. Who was that? Okay, Sylvia. Somebody has murdered my name, Dr. Shogbamu'S Zoo 101. This is not the spelling of my name. Can you kindly correct it? My name's and spelling of my name is Shogbamu. Temidayo, I suppose you're the one writing this. Kindly correct that spelling of my name. Any other question? Ma, you mentioned exomycium. No, I didn't say exomycium, please. That was what I was saying earlier. Please, it's very, very important that you get the correct spelling. Otherwise, if you actually have exomycium as an option in the objective, you would have missed that question. It is endomycium. And as I mentioned on the slide, it is in capital here. And I said that endomycium is the basal lamina that surrounds your muscle fiber or your muscle cell. Is endomycium, not exomycium. So please take note of that. Let me quickly, I see a lot of people's hands up. I just want to quickly attend to those who've put stuff in the slides. Okay, while I go through that, let me see those who are Toluwa logo. Yeah, can you take your question while I go through the chat box?
Morning, ma.
Toluwa, yes, yeah, morning.
Ma, please, can you explain that intercalated disc again? I don't
Okay,
understand that, please.
yeah, okay, all right, I'll do that. And then the Owen says, Ma, please, what is the function of these striations? That's the structure. Remember in our learning objective, one of the things we said, I think the second one was that we should be able to distinguish and describe how the different muscles differ in structure and the function. So the striations are part of the structure of the muscle types that we mentioned have it. And then, yeah, so I think you were saying what was the function of the structure. If you notice, and not that this is confirmed per se, we are basically describing the structure. If you notice, you will see that we mentioned, for example, that our skeletal muscle helps to maintain posture, our cardiac muscles help in contraction. Perhaps, and I'm not saying that the striations could help in that, but there is no typical. The striations are basically part of the anatomy or structure or morphology of the muscle tissue that we have described that has them, right? Tolua logo here, yeah, you were mentioning about intercalated discs. So the intercalated disc, like I said, we only find it in the cardiac muscle. And this is the intercalated, this one that looks like space here. In between the striation that doesn't have any striation, that space there, these white ones, these are the intercalated discs that we have. It's part of the structure, right, of your typical cardiac muscle. I hope I answered that, Toluwa logo. Let me see. There was another person whose hand was up. Okay, there's no other person whose hand is up. Let me go to the chat box now. Functions. Okay, so I've talked about that. Ma, exomycium, I've explained that. Ma, should I read out the question on YouTube? Yeah, Amos, you can read it out or post it in the chat box. What is the function of striation in skeletal and cardiac muscles? I've answered that question, Chinoye. Okay, so I don't see any other question in the chat box. So I'm waiting for Amos to ask the question that were posted on YouTube, I guess. Amos, Amida, I think. Yeah. Morning.
Someone is asking of the cause of dystrophy.
The what?
The causes of dystrophy.
Okay, well, I don't know. It's not part of what we're teaching here. So dystrophy is one of the disease or that associated with the muscle tissues. I don't know what causes it, and it's not part of the content of this course. When you get to College of Medicine and you're talking about etymology of diseases and all of that, I suppose that, or etiology of diseases, you may talk about that, but that's not within the scope of what we're teaching here. I just know that it's a degeneration of the muscle. And so usually it can affect, it can cause stunted growth and things like that. That's all I know. I'm not a doctor. I'm not a medical doctor. I'm a doctor. Okay, any other question?
Okay, please. So the skeletal and cardiac muscle can have multiple nucleus in one of their cells. That's another question.
Yes, as we saw here, yes. It's only in the smooth muscle that we have one cell in and one nucleus in position in the center of the cell. Yeah.
Can you explain the nucleus of the cardiac muscle?
The what?
Nucleus of the cardiac muscle.
Okay. What do you mean by explain the nucleus in terms of showing the structure? This is the structure, and this is the nucleus. Well, if you want me to explain maybe the function or what the nucleus is, remember in a typical cell, you have your nucleus, which is where you have your genetic material. So your DNA, which is responsible for basically reproductive functions and things like that. And then you have your cytoplasm. So in your simple unicellular cells and unicellular animals, the cytoplasm contains different components, which are things that also help in perform the basic functions in the animals, your contractile vacuoles and things like that. So in terms of your direct question, this is the nucleus that we have in the smooth muscle, and this is the cell. Yes. Any other question, Amos, on YouTube?
What is the function of the striation?
I've answered that question. No major function. It's part of the structure or the anatomy of the skeletal and cardiac muscle.
That's all, ma.
Okay. All right. So is there any, I can't see any other question in the chat box, but if you feel I missed out your question on the chat box, please feel free. Okay. Abayomi Ogunleye, please go ahead.
You in the objectives you talked about we learn the organization of the muscles. I don't really get that part in the lecture.
Yes, so the organization of the muscles is what I have shown us in terms of the structure, how they're organized. I mean, if a question were to tell you, let's assume that we are going to write an essay and they tell you to, using well-labeled diagrams alone, differentiate between the structure or organization of the three types of muscle. This is exactly what we are asking you. You can see how the muscle cell here, the components are organized. And structurally, you can see that they're very different from what we have in the cardiac and what we have in the smooth. That's what we're talking about in terms of organization. Any other question? Okay, Ma, please recap the nucleus of the cardiac muscle in terms of arrangement. Yes. So I said the cardiac muscle, we have one nucleus. We have one or two nucleus per cell, and it is arranged, or the nucleus located at the center of the cell, unlike our skeletal muscles where the nucleus are arranged perpendicularly to each other on the cell. That's I answered that question. That was divine. Okay. All right. Is there any other question? We still have about 7 minutes though. Is there any other question? Okay, so if there's no other question, I have some tidbits to give you, some hints to give you, as this will be my last interaction with you, at least for this semester. First, once again, I want to officially welcome you to the University of Lagos, the University of First Choice and the nation's pride. And from the, I know that your set is quite peculiar in terms of it's your set that we're implementing the CCMAS courses for the first time. And like I tell my zoology students for whom I act as course advisor, please, I know a lot of you will be exposed to and you'll be patronized by those who do tutorials outside and all of that. Can I tell you, you are wasting your time and money doing that? Because all the courses and topics we're teaching you from your set, they are completely almost new. Even in terms of the courses, the course load, the course content, they are quite different from what the other sets did. We are implementing CCMAS, so there is nobody that can have information on that. So your best bet is that you attend classes promptly. The other peculiarity about your set is that you are having classes virtually. So a lot of you, at least for your first semester, you've not gotten the feel of being in the university. So take advantage. Some of you are just starting lectures. Please, this is even more critical. Engage with your colleagues. This is not the time to be a loner. You be a loner, you're going to feel like I guarantee you. Because there are a lot of information that have been passed, there are a lot of lectures that may have been done, there are a lot of course contents or course material that may have been given or sent, which you may not have been able to access. I don't know if some of these things, if you all have access to LMS now, that is the learning management system, the university, which would usually have a one-stop place for that. If you don't, it means that you will need to ensure that you liaise with your colleagues, not the unserious one. Identify the course rep or course assistant, somebody who's always getting information. And if you cannot, please, all the lecturers, even though you are being taught virtually, we are all available and accessible to you. Not that 1,000 of you should be coming to my office. But if you go to the department and request for information or you talk to the sub-dean, they will oblige you. And I'm aware that there is a platform where all you students are, where the sub-dean interacts with you. Please, don't get it twisted. We have rules in the university. Hopefully you have come in on merit, you didn't come in through the back door. If you perform or you participate in any misconduct in the forthcoming exams, both practicals and the main exams, you are going to face the brunt of the law. And there's serious penalties, from rustication to expulsion. Using any means. I know now, all of you know how to use AI and all of that, how to get some of those things. My name is Temitope Shogbamu. Please note my name and my department. If I find my lecture slides, my notes, my video online, circulating online, and not just for this group, I am going to hold Temidayo responsible and all the course reps, and I will ensure you face the law. My name is Temitope Shogbamu. I'm a senior lecturer in zoology department. So please, if you are peddling lecturers' courses, you are working with some study centers or tutorial centers, and they are telling you to be sharing the slides or whatever with them so that they can be selling it on whatever platform, please don't peddle my own. I'm saying it clearly now, and you can see this is on YouTube. Don't peddle my own. If you do that, it's a very serious offense. And it can even get you expelled from the school. I can guarantee you that. So don't let anybody use you, and don't use yourself. If your plan is to come into the university and come and take advantage of lecturers, they are not here to study, you better rewire yourself. At least not for my own course. Maybe the other lecturers are okay, not for my own course. So I just want to warn you all, but I also want to encourage you, particularly for those of you going to College of Medicine. You don't have time to play. It is very, very competitive. You must get A, and guess what? Even if you have A, it's not a guarantee that you're going to College of Medicine. So it means that you have to be among the top best. It's a competition. It's not everybody that will go. It's not a course. So it means that you have to double up. work hard. You cannot play like others are playing. Not to say that the others should play. Like I tell my students, you should aim to have nothing less than a B for all your courses. You have all sorts of resources available to you. You are doing the LMS here, you have course slides shared, you can get additional materials, from online and all that. But please, while there's so many materials out there, some of them can take you outside of a course focus. And then you are reading much more than is expected. This is still year one. Stay within the course synopsis, stay within the expectations, and I'm sure that we lecturers would have given you that. If none of the other courses have given you, please feel free to ask. We lecturers don't bite. We may be overwhelmed by multiple requests. If you come in respectfully and you ask us that you would like to get the course content just so that you're able to read alongside and all of that, I'm sure nobody will say no to you. We'll all oblige you because we're not going to get anything from you failing. And I can guarantee you, if you also pass, we can all get 100% pass. It's not a must that some students must fail. No, we don't hoard marks here. If you all pass, you will get your marks. I'm sure you've seen or you have heard the news of students that graduated with 5.0 or 5.0 in Unilag. So it means that it is possible. Whether you are going to College of Medicine or you're staying back, you're doing zoology, cell biology, it is very possible for you to hit that mark. And that should be your aim. So that when you're aiming for the very best, let's say anything happens, you come to maybe 4.5 or something, not anything less. So we are at the top of the hour. I want to wish you all the best, and I'll see you all in the exams for your first semester courses. Have a nice day, everybody.
Thank you, ma'am. And you too, ma'am.
Thank you very much, ma'am.